My name is Betsiti Araya, and I am an undergraduate research assistant at the Neuronal Networks and Interfaces Laboratory at UTD. The project I am presenting is the behavioral analysis of ultrathin microelectrode arrays. Intracortical microelectrode arrays, or what I'll be referring to as probes in this presentation, are devices that are implanted into the brain to record or stimulate neuronal activity. These probes have enabled great strides in investigating the nervous system and have been applied in clinical scenarios such as brain-machine interfaces. However, conventional probes are made up of high modulus materials such as silicone that have drawbacks associated with their size and flexibility. These drawbacks may induce considerable foreign body responses that tend to have adverse effects on the motor function and behavior of animals with implanted probes. In this study, probes made out of amorphous silicone carbide, or ASIC, are compared to the commercially available and conventional neuronexus probes that are made out of silicone. The comparison is in terms of behavioral deficits caused by probe implantation in the motor cortex of animals. The ASIC probes are of special interest because they are fabricated with ultra-thin dimensions that result in better flexibility. These enhanced mechanical properties are expected to reduce the response resulting from the probe insertion. The in vivo experimental design is 16 weeks long with 8 ASIC and 8 neuronexus rats. However, this presentation covers the first 6 weeks of the study, including only 6 ASIC and 4 neuronexus rats. The probes were implanted in the left motor cortex, as shown in Figure 2, and after implantation, the rats were recorded weekly on the cylinder test for 3 minutes and trained weekly on the knob supination task for 30 minutes. The first behavioral measure is the cylinder test, which evaluates changes in the limb use asymmetry and overall exploratory behavior. As shown in Figure 4, the rat is placed in a clear cylinder where a camera is placed underneath to record the paw usage of the rat. After a 3-minute recording, the video is analyzed by manually counting the number of times the left paw, right paw, or both paws were used to initially contact the wall of the cylinder. The results from the cylinder test showed that the ASIC rats returned to their baseline level of exploration at a faster rate than the neuronexus rats, and this supports the hypothesis that ASIC devices inflict less foreign body response and allow for faster recovery. Unexpectedly, both right and left paw dominant rats of both ASIC and neuronexus groups showed significant changes in their paw preference over time. For more conclusive results, changes will be observed over the remaining 10 weeks of the study. However, the preliminary results may show that the probes do not affect paw preference. The probes may only affect complex motor movements such as the knob supination task and not relatively more passive motor movements such as cylinder paw contacts. The second behavioral measure is the knob supination task, which evaluates the ability of the rats to learn and perform complex motor movements such as reaching, grasping, and rotating a knob. The differences between the ASIC and the neuronexus rats were mainly observed in the learning of this task, where the ASIC rats were able to reach the final stages of training while the neuronexus rats had not completed the first stage after initial habituation. This showed that ASIC rats learned at a considerably faster rate than the neuronexus rats and that complex motor movements are less impacted by the ASIC probes. In the future, the knob supination task can be used to record neuronal activity as the rats learn the task of reaching, grasping, and rotating a knob, and this data can allow for the ASIC probes to be used in creating a motor decoding algorithm that can predict motor kinematics for controlling neuroprosthesis.